My speech will be about hyperspectral imaging. Introduce you our program, which we have. The program, at first I'm a specialist of biomedical equipment. Now I work at the institute named for Physiology Institute at the name of Orbeli. I would like to inform you a few words about the program we are implementing. Founded by Chan Zuckerberg Initiative, the Orbeli Institute of Physiology is implementing a program to create a network of biomedical imaging professionals who will have the opportunity to share experience, participate in network-based courses, and most importantly, network research equipment. The main purpose of this network is to promote research in biomedical imaging, to provide opportunities for researchers, and there are several goals which you will have. At first, to form a regional, our goals is to form a regional biomedical imaging society with a dedicated website, to create a database of bioimaging equipment and expertise available for collaborative projects and trainings. It's our website, and if you want to be a member of the society, you can register there and you will have all opportunities to be being network member. The benefits to be our member, it's cost-free attendance to schedule or demand training sessions. You can have trainings about microspectral imaging, about confocal microscopy and cell staining, and which you will choose. The registration in the website is free. There are also travel awards to attend the annual BIO-SEE meeting, personal advisory sessions, and design and analysis of imaging experiments, etc. Well, at first, I'd like to answer to the question, what is light? Because we speak about hyperspectral imaging, and imaging is based on physical, what is light. Light, as I said, is an electromagnetic wave, just like any other wave. Visible light has its own characteristics, of which the wavelength is significant for us at the moment. It means the wavelength is the most significant part. Well, what is electromagnetic radiation? As you see in the picture, electromagnetic waves are very diverse, and depending on their wavelength, there are radio waves. You see here, radio waves. There are microwaves, infrared waves, ultraviolet, visible spectrum of light, X-ray, and gamma rays. Here you see comparison of each type of wavelength with size of different objects. For example, radio waves you can compare with this kind of buildings, or humans, or butterflies. It's only we can detect by our eyes visible light. It's very narrow range of electromagnetic spectrum. Visible spectrum starts from 400 nanometers to 700 nanometers. This range of electromagnetic waves we see as colors, but actually in nature there are no colors, but color is a perception of our eyes and our brain. Also, here you can see the sources of different electromagnetic waves. The radio waves, the source is AC power, and for television. For telephone works microwaves and satellites. For the source of light, visible light, is sun and bulb, as you see. Well, and I'd like to ask a question about vision. How our vision works. Actually, the spectrum of electromagnetic waves is very large, but a very small range of the whole spectrum causes a sensation of effect in humans. Is the range of visible light, which by stimulating specialized cells of the retina of the eyes creates a sense of color. These retinal cells convert the light energy into biopotentials that travel through the nerve fibers to the brain, where they are processed. Yes, that's it. That is, electromagnetic waves waves do not actually have colors, as I mentioned. Every living organism sees the same spectrum of visible light in different colors. The perception of colors depends on the light sensing cells in the retina. The brain that analyzes the information received by those light-sensitive cells. For example, there are two types of receptors of human retina: color sensitive and light sensitive. Here is the light sensitive, and there is color sensitive, which you see in colors. Light sensitive cells are able to be stimulated by a small amount of light. In dark conditions, they are able to see the objects. We are able to see the objects, but we can't have a perception of colors, and it helps us to orientate in space. The second type of cells are color sensitive. They are able to be stimulated by higher intensity of light, under the influence of which the color sensitive cells are stimulated, giving a sense of color. Here you can see the number of these pigments which you have seen are various from animal to animal. Humans have three color receptor cells: blue, red, and green. For example, mantis shrimp has the most light-sensitive cells. Most light-sensitive cells. It has 12 different types of cells, which combines 12 different range of electromagnetic spectrum. And the human with only 3 color cells responsible for transmits information to brain. Our brain is a very powerful analytical machine, but it has some limitations. First limitation is subjectivity. Every human brain develops in different directions and on a different genetic basis, so each brain interprets color differently. Different people interpret different colors looking at the same image. Subjective perception of colors does not allow people to talk about particular color shade because saying green, all men, mean different shades. So there was a need to differentiate between different shades by coding them. The second limitation is the very narrow range of visible light from 400 nanometers to 700 nanometers. The third limitation is the need for a large amount of light for colors sensitive. When white light falls on an object, it absorbs all the spectra and reflects the spectrum it has not absorbed. So we see the reflected light, which is intense enough to stimulate the cells in the retina. Sensitivity of these receptors is limited. It means that these receptors need much light, as I said, for providing color vision, and when it is dark, we can see colors, but we can see only shapes of objects. But for biologists, there is a more important phenomenon that the naked eye does not see. This phenomenon causes autofluorescence. Actually, fluorescence is the phenomenon when we illuminate an object with, for example, a spectrum of blue light, receiving a light of a longer wavelength spectrum. However, fluorescence has a very low intensity and it lasts about picoseconds, and human eye can't detect it. Various technologies have been created to get out of the listed limitations and get more informative images, one of which we will talk about now. The technology called Hyperspectral Imaging. Actually, what it is, this technology has been used in many fields, but now aware about this technology and about the opportunity which can provide this technology. Hyperspectral imaging is a technology which combines imaging with spectroscopy. Every chemical substance has its own specific optical characteristics, and with the help of spectroscopy we can quantify optical characteristics of each biomedical, biochemical substance, and machine can differentiate healthy and blemished parts of the surface of paper. Here we can see image, which has the intensity of the light reported. This is a grayscale image. This one, grayscale. Then you can see with RGB colors recorded by camera or seen by human eye. And the third is example of image composed by hyperspectral imaging. Here we see an image of same apple, but the help of infrared range of biomixing of spectral differences between healthy and blemished parts on the surface of apple because of their different biochemical composition. So hyperspectral imaging allows us to overcome the listed limitations of human vision. The first limitation is subjectivity. As mentioned before, human vision and brain is subjective, and human can't save the image, can't monitor, can't quantify, and can't give the information which will have particular spectrum. Also, human vision depends on physiological conditions which vary every other hour. Hyperspectral imaging allows us to quantify, save, compare, monitor images and its objective for everyone. Second limitation is only few types of photoreceptors, red, green, blue, and with the help of these three photoreceptors, human can differentiate up to 10 million shades of color by mixing them together. Hyperspectral imaging allows us to have detectors for every spectrum of visible light and near visible spectrums. Hyperspectral imaging technology usually has a set of filters which it puts in front of individual wavelengths that stack of images then go to the computer, which then extract from each individual pixel of the image the entire spectra, and the spectra gets sorted by mathematical algorithms, and as a result you can ascribe different colors to different pixels of the image which have different spectras. Also, you can make reports about interested objects. Third limitation is limited spectral range of human eye. Hyperspectral imaging allows us to detect more spectrums behind visible light spectrums. It detects infrared and UV light, depends on particular machine. Sensitivity of these receptors is limited. It means that these receptors need much light for providing colors vision, and when it's dark you can't see. Hyperspectral imaging allows to illuminate detectors for a long time, and they can collect required quantity of light and analyze the spectrums which can do human eye. So, having ability to quantify colors provides a lot of useful information, particularly when you want to monitor, compare, and report changes in time. Hyperspectral imaging has several advantages and disadvantages. First advantage is that it's non-invasive and camera detects only returning light of the object. Because we just analyzing returning light, the area of infrared and UV, which also can be used, are not in the dangerous domain. We don't introduce any damaging or radiation. We don't need any additional dyes or other order to distinguish the objects. We can reveal very small differences in color, something which can appear in front of human eye. Spatial resolution of hyperspectral imaging is high. It's basically limited by the wavelength of light. It's about half of the wavelength of the visible range, about 200 from 200 to 300 nanometers. And disadvantages, because we are using light reflected from object to the camera, we cannot really see inside the object. So we limited acquiring the information from the surfaces. In order this technology to be useful, we must know application-specific ranges where these differences can be amplified. You can acquire hyperspectral cube in 4 different ways. At first, you can do it point-by-point scan. You can do line scan when you're acquiring line by line, and it's called push-pull scanning. You can do wavelength scan, and finally there is more developed method of snapshot acquisition using spatial cameras, which allows to focus every image to several areas of sensor. Therefore, the entire cube can be acquired simultaneously. Depends on these 4 types of scanning technique. There are 4 types of cameras. Each of them has limitations and advantages. The point scanning has very high spectral resolution, but it works slow because it has to move point by point. The push-broom scanning is mainly using for objects where you have conveyor belt and the camera above allows to monitor the passing objects line by line, or it can mount it on drone or airplane when it flies over the field, and to acquire that image sensor is moving across the sample. Spectral scanning is the one which used mostly in biology and medicine. By this method, you acquire spatial image by individual wavelength, then filters changes physically by rotating the filters. You have tunable filters for each image is acquired at individual setup wavelengths for the hyperspectral cube. And finally, there is new technological development which allows to acquire a hyperspectral cube at once. In this case, there are so-called mosaic sensors where you can project multiple wavelengths into different areas of sensor. The method works very quickly. There is a wide range of applications of this technology. It has been used in satellites, in military flying above field. It can recognize soldiers' uniforms and recognize which army they belong to. In agriculture, it is used to understand whether the specific areas of the crop needs fertilizer or additional water supply or pesticides need to be added. The agriculture has a wide range of applications. Also, hyperspectral imaging has application in recycling to differentiate many types of plastic with the help of UV light illumination. With the help of hyperspectral imaging, also they sort different kinds of fruits and vegetables in health and conditions and with problems. And finally, hyperspectral imaging you can apply in medicine. Changes of color on the skin you can observe and differentiate with hyperspectral scanning. You can analyze every surface of human body. You can use it in surgery, in pathology, in oncology. You can observe changes in human body. Anyway, you want to, you can do scan and mix the wavelengths, see changes, quantify and save them. Because of the rapid decrease in price, hyperspectral cameras start to be available for researchers, and this graph shows the amount of applications in color. Here we can see the application of hyperspectral imaging in recycling of plastic. If human eye can hardly differentiate different types of plastic, hyperspectral camera is distinguish them easily, as you see in the picture. Hyperspectral imaging is promising, but still young technology in medicine. There is a term called medical hyperspectral imaging. It is a young technology, but already it has a wide range of applications. For example, cancer detection, cardiology, neurosurgery, pathology, etc. There is a perspective of quantitative measures of microvascular perfusion values in the skin. Hyperspectral imaging devices identify unique spectral fingerprints of oxygenated and deoxygenated hemoglobin in the surface of microvasculature. Establishing value ranges for health patients without vascular complications will subsequently help standardize assessments for perfusion detection. In particular, diabetics who are prone to vascular calcifications of lower extremity wounds may benefit. The number of people who have been diagnosed with diabetes has nearly quadrupled in the past 25 years worldwide. Hyperspectral imaging is a novel imaging modality that evaluates level of oxygen and deoxygenated hemoglobin within a tissue plane to be depth of 1 to 2 millimeters beneath the surface of the skin, based on penetration of near-infrared or visible light. Oxygenated and deoxygenated hemoglobin have unique spectral fingerprints and that have been identified in the literature. Capture and analysis of these reflected wavelengths can quantify blood hemoglobin, giving this technology an advantage over Doppler and ultrasound angiography. The imaging devices Hyperview, here it is. Hyperview are portable, non-invasive cameras that combine light arrays, high pixel density, photoreceptors, and analytical softwares to measure microvascular perfusion values. It uses near-infrared light with wavelength of approximately 830 and 716 nanometers to quantify oxygenated and deoxygenated hemoglobin in tissue. Devices provided quantitative measures of hemoglobin in localized areas of the body. The real-time visual representation of microvascular perfusion has extensive implication in medical and surgery. This is a camera has medical application using infrared light spectrum. Camera detects the light emitted by surface of skin. This kind of technology isn't so widespread, but you can, with the future doctors, will be used and make healthcare better and safety. With the help of HIS, you can analyze and by any biological surface you want. Speaking about microscopy, you can detect it not only on the feet, you can use it for detecting retina, brain, skin, in open surgery, anywhere with blood vessels. In this picture, you see the open surface of brain. When you look at the surface of brain, you can distinguish the parts of the brain which have circulatory disorders. But as I mentioned before, as the biochemical composition of healthy tissue and ischemic tissue are different, camera can easily detect these differences and quantify the optical spectrum. It allows us to see the differences easily. Is another interesting application of hyperspectral imaging. With the help of hyperspectral imaging, our arithmologists detect the lesions of endocardial tissue after radiofrequency ablation. Radiofrequency ablation is a minimal invasive technique that shrinks the size of tumors, nodules, or growth in the body. When doctors do the RF, they need to visualize lesion after RF ablation, but as you see now, endocardial tissue is white and naked eye hardly can recognize and distinguish the lesions. Well, HSI helps come here. With camera, they make hyperstack of images where you can visualize the ablated lesions and in outcome you see the results which treatment you have done in heart. This was the pig's heart. Now you see the same reflection in human heart. Here you see the white surface of the endocardium, but there are some lesions after RF ablation. Here you can see how spectral degree correlates with lesion depth change. HSI lets you to make correlations with different degrees of lesions. HSI can reveal multiple features of tissue types, blood vessels, fat, collagen, etc., which couldn't do our eye. And so, dear participants, I would like to conclude by passing on thoughts to you. You are doctors of the century during which we will communicate with large amount of technologies. During your career, you will face many problems that can be solved by developing new technologies that will make your hard work easier and more efficient. HSI is one of them, and you can generate many ideas for applying it in medicine and make it better. It allows you to make everything more visible. Think about it. If you have any idea about application of this technology in medicine, and you want to make research in it, I will help you in this work. And it's over. Thank you. Any questions?
So you told that fluorescent light cannot be seen through our eyes.
Yes. We can't detect it. Fluorescence. Yes.
There are some toys and some few things which these are fluorescent. We could see.
It's called luminescence. This is different. Fluorescence lasts a very short time. It's billions of 1 second, which we can see. It's called luminescence, and it lasts longer. It's used, for example, here in the watch I have. When I illuminate it with light, I can see. It's called luminescence. But fluorescence is different kind of light. When we excite a specimen or any other color which have fluorescence, it lasts very short and our eyes can detect it very fast. Like lightning, but very short.
Yeah. So then one more thing. In hyperspectral imaging, which spectrum or which wavelength of light are you using?
It can detect wavelengths starting with UV till infrared. Infrared, we can see infrared. It's more than our visible spectrum, but but hyperspectral cube, hyperspectral camera can detect it, can see it. It's about more than 700 nanometers, about 900, 1000, 1100, and so on. The range of infrared light.
And one more thing, could you please show that images, the fluorescent images of in medical application? And even
here,
you told that in hyperspectral imaging, we can't see the inside of an object.
Yeah.
But the heart and all are inside our body. Then how do we see that?
Heart inside,
Yeah.
but they extract heart from pig or human and see. And there are intravascular catheters which goes through vein. They using the optofibers, air optofibers, which arrhythmologists and cardio surgeons uses. They put it. The optofibers goes through veins to heart. It's endovascular surgery. They uses them and they eliminate heart tissue inside of endocardium, and by this reflected light they can analyze the optofibers which in heart goes through hyperspectral camera, and camera sees the inside of the heart. It works like this. Or here.
Yeah, here. So it is the hyperspectral imaging, right? But I think it is the image of same group. Is it the image of taken at same time or different times and different? Why is it so different? Because in the first image we would see orange, red, all color, and in the second image we see only blue color. Why is it so different?
Differences is every biochemical substance has its own optical characteristic. For example, water and wine has different interactions with light. For example, light goes through water, but through wine, no. It means that every biochemical substance has its own wavelengths, which needs to reflect it. Here you can see oxygenated and deoxygenated hemoglobin, which is blue is deoxygenated hemoglobin, and which is red is oxygenated. And here you can see differences. Here they detected the saturation of here they detected microvasculatory microcirculatory process. For example, the food which blue has problems with circulation, with blood circulation, and which is red, the blood circulation starts to normalize. Yes, and the different parts, the difference in these pictures are the condition of the food. For example, different circulation conditions, they make these differences.
So we can only use this on substances whose optical properties are already known?
Yes, but it's easy to detect new substances. For example, we were doing such experiments in Hungary. It's very easy to do. You should put it a spectral photometry. You should these substances you should put on the camera inside, in front of camera, and the camera has its own program computer. You make your algorithms, which wavelengths you want to detect, and then camera starts. You illuminate it with white light, which has all spectrums, and which light it reflects. This is the spectrum of that substance, and the camera can detect it, and camera gives us the correlations, and we can measure it. No, any other question?
Do I have just a few questions? First, currently is it used worldwide in medical field?
It's very new in medical. Actually, this technology is invented in NASA in 1972. They used it in satellites to detect the surface of Earth, and their as there are snow, there are cloudy weather and so on, it's better to analyze with hyperspectral methodology, not RGB, because it gives more information. They use it in satellites till now. In army, they use it to detect the soldiers with theirs or others. They use it.
Agriculture.
Yes, in agriculture, and medical application is new. That's why I want to introduce you with the technology for to find a better application for this technology. And I think it can be many applications in medicine for this technology.
Do you think it would be like most useful in dermatological issues? Like, as it's safe for another question, if it's safe and no like radiation harms, so people may use like the device at home or some like I don't know, maybe like practical, like people can use it not only in hospitals, and they can check. For example, there are some malignant process. For example, they are like benign moles can be cancerous, so they can check at home or somewhere if there is no radiation injury. So do you think it will be such practical, like people can use it at home? Or
there is such practice, but there are some tiny cameras which you can put in front of your smartphone, and your camera becomes hyperspectral camera. Actually, it's a tiny camera, and it's for personal users. People buy these cameras. It's not widespread. It starts to be widespread now, and it uses for cancer detection. Because actually what we see on skin, we can't differentiate many types of cancer and the stages of cancer, but these correlations, we can make these correlations, and it can differentiate more narrow spectrums which we can't see. And these narrow spectrums which we detect, we can save them, and we can make a library from that correlations. For example, I know that melanoma in this stage, in this part of the skin, in this type of skin, has such kind of correlations. And by that practice, you can easily detect which like your correlations. I have such a program, and the aim of this program to make such libraries which can have many types of correlations, and we can make AI-based technology which can make these correlations more faster and better, and we work on it. It's one of the applications of this technology.
Where can you buy these cameras?
Excuse me?
Where can we buy these cameras? You said they are already available.
It starts about $20,000 starts, and go higher and higher and higher. And the one of these types of cameras is called Nuance. You can check it in Google. Which we use. It's about $60,000, but there are better cameras. If you type hyperspectral camera, there will be so many. Already it's so many.
one more question, Gevorg. Like, as I understood, like hyperspectral imaging is about better seeing something. About medical applications, like how can we know the hyperspectral characteristics for certain conditions? Like, what is hyperspectral characteristic of melanoma? Like, we know that like a biopsy, not in melanoma, but in other cancer, has a definite diagnostic option because we have literature that if it looks like microscopically in this way, this is this cancer. Like, when we are going to use like hyperspectral imaging diagnosis, do we have some literature that this one in us hyperspectral means this condition, these things? How we can overcome this problem?
There are some researches about this question. If you want to detect some kind of cancer, at first you need to do biopsy, for example. When you do biopsy, or if the cancer is on skin, and you already diagnose it. It's melanoma, it is melanoma, in such stage, such kind of special, its stage. You put it in front of camera, and the camera makes these correlations. You should do more such kind of experiments and make this database, and then to write a program which can make these
calculations.
Yes, calculations, and then it can making by this calculation can say, for example, what I see about 95% is melanoma in this stage.
Sorry, is it about machine learning technologies? And one question I have actually, could you please discuss a little bit more about the uses in microscopy? Because microscopically we have seen a lot of different applications, but interestingly enough, there are some also uses of the hyperspectral imaging in microscopical images, and what are its advantages over more conventional and high resolution microscopic techniques?
Actually, there are not so many differences between microscopy and traditional objectives. Microscopical way to detect it, its advantage that we can use colors which can do fluorescence, and hyperspectral imaging, hyperspectral camera can detect fluorescence more better. But which image you put in front of camera, it analyzes the same way. If you put it in front of microscope is also objectives which only make magnification, but the image is the same type or the same size image goes through camera and detects by this detector.
So why is it not so widespread in medicine today? Like, what's holding it back? Is it because people don't know about it so much?
Because people don't know.
Yeah. And if there are institutions who will adopt this new technology, in what price range would each test be?
A test?
Oh yeah, with this.
Actually, we are doing such program for free. I mean, if you are interested in it, and if you want to do some research on it, being the member of BIO-SEE Society, to register a website. You can write us, we can connect, and you can use this camera however we want. And also we can do consultation about it, how you can use it for your research program. It's absolutely free.
It might be useful for everyone. That's why I would like to. You
I
can.
would like to know here in BIO-SEE, the website UTMA, some of the courses are written, they are prepaid or paid per course. Yes, I would like to know which one is free or how we can become a member of BIO-SEE, and after that, will all the courses be free or no?
Actually, I don't know why they're written so, but our courses are very free.
Are free for the members of the BIO-SEE.
Yes.
Individuals outside
You
the
can
BIO-SEE.
go to membership. You can there register in website, and then you can write us.
And the course is free. Okay, thank you.
So
Thank you very much.
thank you.
Thank you very much.